Hi, I'm Kendall Kleckner, and in my group I have Connor Everett, Troy Damaris, and Sarah Swanson. In this video, we will be explaining what gene expression is and what happens during it, as well as each part of the process and how they contribute to gene expression as a whole. We will also go more into detail about how translation works and what parts contribute to the process. We will do this with a model we have created. What is gene expression? Gene expression is the translation of coded information in a gene into a protein that are present and operating in the cell. These expressed genes include genes that have been transcribed into mRNA and then having it translated into a protein. The gene is expressed through translation, RNA editing, and transcription. Through the process of transcription, it starts with the gene, and the RNA polymerase that binds to that gene synthesizes pre-mRNA. That new pre-mRNA is edited by spliceosomes and snRNP. It removes the non-coded information, which are called introns, and bind the coded sequences, which are called exons. Once this process is done and we now have a functional mRNA, that mRNA has two caps that are added as protection when traveling to the ribosome for translation. This process allows the mRNA to become a protein with the help of amino acid and tRNA. Once that protein is made, it is released into the cytoplasm.
A ribosome is a molecule found within all living cells that serves as a site of biological protein synthesis. Ribosomes link amino acids together in the order specified by messenger RNA molecules. A ribosome is made up of a large ribosomal subunit and a small ribosomal subunit and has an mRNA strand running between the two. Within the ribosome is the A site, which is where the tRNA binds to the ribosome. This tRNA reads the mRNA strand and finds the correct protein for the codon. The tRNA then moves to the P site, where the proteins that the tRNA brings are attaching with peptide bonds to create a polypeptide chain. Lastly, the tRNA arrives at the E site after it has dropped off its amino acids and exits the ribosome. This process of the tRNA moving through the ribosome creates a polypeptide chain, or the primary structure of a protein. In gene expression, DNA, which is in the nucleus, is the polymer that gives the template for the entire process of gene expression. It is what is used to create the mRNA, which helps translation occur, and without the process would not be able to occur. So it is a pretty major part of gene expression, and without it, the whole process would be impossible. Messenger RNA, which is this strand right here, also plays a big role in gene expression, and that is to link genes to proteins. These proteins that are created help our body function and move. Transfer RNA, or tRNA, holds a central place in protein synthesis by interpreting the genetic information stored in DNA into the amino acid sequence of protein, thus functioning as adapter molecules. Lastly, ribosomal RNA, which creates ribosomes, is part of a ribosome, which ultimately make the whole process of translation possible and creating a polypeptide chains. A small ribosomal subunit attaches to the start of the mRNA sequence. Then the tRNA molecule goes through the A site carrying an amino acid.
The molecule that is brought in by the tRNA through the A site is called methionine, and that's called the start codon. And the anticodon on that tRNA is going to bind with the codon on the mRNA to start the sequence. Then this large ribosomal subunit binds to form the complete initiation complex.
Elongation occurs in the P site of a ribosome, where it creates the polypeptide chain. After it creates the polypeptide chain, it will go to the E site.
After initiation, elongation occurs with this anticodon, this tRNA up here, and this codon. The anticodon bonds to the codon, and an amino acid is made. After this occurs a couple times, peptide bonds are formed in between amino acids and forms this polypeptide chain.
In termination, the tRNA enters the ribosome in the A site, where tRNA molecules bind to the stop codons, so the peptide chain becomes hydrolyzed, thus releasing the polypeptide into the cytoplasm, starting off a series of events that separates the tRNA from the ribosome through the E site and creates finished polypeptide chains.
In the process of termination, the anticodon attaches to the stop codon, which then stops the formation of a forming polypeptide bond, leaving a polypeptide chain to be released as a protein.